Good evening, everyone, and thanks for Shares for letting me present tonight. This is our disclaimer statement. Obviously, we're a public company, so I need to show you this, but moving swiftly on. A slight change in pace. ReNeuron, for those of you that are unfamiliar with us, we are a cell therapy business, so we're harnessing the therapeutic potential of stem cells. So we're a cell-based drug developer, if you will. We're developing non-patient specific or allogeneic stem cell therapies from two core assets, which I'll come on to in a second. We have a retinal cell line and a neural or brain stem cell line. And those have formed the basis of our clinical programs using cells purely as therapeutics. We have a couple of other strings to our bow as well these days, which I'll come to. Our lead cell therapy program is in phase 2, targeting retinitis pigmentosa, a blindness-causing disease. And again, I'll come back to that in some detail in the presentation. Importantly, that's where we believe there is licensing potential once we garner further data over the next 12 months or so. And again, I'll come back to what that really means for us in more detail. And those other strings to the bow that I mentioned really revolve around our exosome and induced pluripotent stem cell research platforms. And again, I'll leave it there and come back to those in more detail as we go through the presentation. So the 4 platforms we have, a little bit more detail on each of them here before we get into the programs in more detail individually. So our HRPC platform, HRPC stands for human retinal progenitor cells. Those are the retinal cells that we're using to target retinal diseases. And those cells have the ability to differentiate into the photoreceptors that are lost in these retinal diseases, and also have the ability to engraft into the retina. And that's if you like, the therapeutic hypothesis for why we believe these cells could play a key role in addressing the deficits associated with retinal degenerative diseases. Importantly, as a therapeutic, we formulate these cells in a cryopreserved formulation, which allows for ready shippage and storage and use at the hospital site when required. So it's not only about garnering clinical data, it's also providing the means to manufacture or scale these cells at a cost of goods that are going to be relevant going forward. As I mentioned, we have this program's in phase 2, and we have positive data in phase 2 in retinitis pigmentosa, and I'll come back to those data in the presentation as well. This program is also partnered with Fosun Pharma for the Chinese market, as indeed is our CTX platform as well, which I'll come to in a second. Our 2 research platforms I mentioned, exosomes and iPSCs. Exosomes we can harvest from our neural stem cell population, and exosomes I suppose are an increasingly interesting field, and something that where there's been quite a lot of commercial interest actually, and I'll come back to that to evidence that a little bit later in the presentation. But importantly, what we can do with our exosomes is load them with RNAs, proteins, and other molecules of interest that have therapeutic potential. So in other words, we're using our exosome platform as a potential delivery system for delivering novel drugs. Importantly, we know our exosomes can cross the blood-brain barrier. That's vital if one's addressing diseases of the brain, for instance, and that's a key characteristic which we believe gives us an edge in this field. Our iPSC platform, again, iPSC stands for induced pluripotent stem cells. And again, I'll come back to what that means when I get to this program towards the end. But again, this is technology that really was a spin out from the development of our CTX neural stem cell line, and actually plays to some very interesting technology that was developed a few years back, actually, to turn, if you like, adult stem cells back into an embryonic stem cell state, hence the term induced pluripotent stem cells. And we've been able to achieve this with our CTX cells. And that opens up further opportunities in terms of generating new cell lines for therapeutic use, and from those lines developing exosomes, again based on non-neural stem cells. So it gives us potential new therapeutic candidates going forward. Finally, our CTX cell platform, again, this is another pure cell line developed as a therapeutic, being targeted against stroke disability. We've been working with this program for a number of years. Importantly, we have further data preclinically to show that our CTX cells have merit in other CNS diseases, potentially including Huntington's disease. And again, this particular program is partnered with Fosun Pharma for the Chinese market. And I'll come back to CTX again briefly towards the end. So our development pipeline is listed here. So again, 2 programs using cells as therapeutics in phase 2, with further data expected with the retinitis pigmentosa program over the next 12 to 18 months as we progress that study further. And I'll come back to that in a second. And our exosome and iPSC platforms at an earlier stage, these are very much in the research or preclinical stage. And the idea here is to work collaboratively with third parties to help deliver their drugs using our exosomes predominantly. And we've signed a couple of deals so far, 2 or 3 actually, and some more in the pipeline. And again, we can target our exosomes and iPSCs at a number of therapeutic targets, both within the brain and beyond, as you see here, including COVID-19. And I'll come back to that when I talk a little bit more about exosomes. But before that, let me just dive in a little bit more deeply into the retinal progenitor cell program. As I mentioned, our HRPC cells are an allogeneic or non-patient specific cell-based therapeutic candidate targeting retinal diseases generally. Again, based on these cells' ability to differentiate into functional photoreceptors and integrate into the retinal layers at the back of the eye. And we've shown this preclinically. We also know that these cells have an ability to engender trophic support at the back of the eye as well. We regard this as a platform, so we think it has potential across a range of retinal diseases. We're initially targeting retinitis pigmentosa, and to that extent, that particular program has orphan drug designation in Europe and also in the US. And in the US, our program against retinitis pigmentosa also has FDA fast track designation. Importantly, as I mentioned earlier, we have to generate clinical data to show that the treatment is safe and effective, but we also need to ensure that we can manufacture and scale these cells with ease at a cost of goods that's going to be relevant. And we've been working with collaborators across a number of years, both at Harvard and also at Moorfields at UCL, to optimize the way that we manufacture and scale these cells under good manufacturing practice or GMP conditions. And as I mentioned earlier, we now have a cryopreserved formulation of these cells under a long shelf life, which of course enables easy shippage and storage worldwide. So something that can be used when required, and therefore perhaps is more akin to a more standard biologic drug, an antibody, for instance, even though these are actually living cell candidates. So the first target is retinitis pigmentosa, as I mentioned, for our HRPC platform. RP is inherited degenerative eye disease. It's a relatively large orphan disease, 1 in 4,000 incidents in the US and beyond. And actually RP as a disease condition is associated with a number of individual gene mutations, which collectively form the retinitis pigmentosa as a disease. And there's only one treatment available at the moment that's been approved, and that's for one of those particular gene defects, RPE65. So importantly, what we're doing is looking at a cell-based approach that is gene independent, with a view to addressing those deficits, either slowing down or even arresting the sight loss that you see in these patients. And that's the essence of our HRPCs against this particular disease. That's what we're looking to achieve. And RP as a disease, again, the panels on the right here show you how this disease manifests. It's essentially an initial loss of peripheral vision, leading ultimately, and what can be over quite a large number of years, ultimately to blindness, unfortunately, in these patients. So having got into the clinic in the US a few years back, we've now completed a phase 1 study with our cells in RP patients. These patients had very poor visual potential. There were 12 of them, and during the course of this phase 1 study, we changed the formulation or optimized it from a fresh formulation with no more than a few hours of shelf life to a cryopreserved formulation with a much longer shelf life. And that then led us into a phase 2 study, or actually the phase 2 element of this combined phase 1-2 protocol. And in that part of the study, we've now treated 10 patients with established RP. These patients had better visual potential because, of course, we were monitoring these patients both for safety and also for secondary measures on efficacy, including things like visual acuity, visual field, and so on. The study so far has been run in 2 sites in the US, listed here, but we now have regulatory approval in the US and the UK to extend this study into further sites, and I'll come back to that in a moment. We've been very encouraged and excited actually by the data we've seen so far coming from this study, and we've been able to keep the market updated in terms of how this study's going because it is an open label study. It's not a blinded placebo-controlled study. What we're doing here is treating patients against the baseline level of sight, measuring both the treated eye and the untreated eye, if you like, as a quasi-control. So again, I emphasize it's not a true placebo control, but it gives us some ability to monitor the putative efficacy of this treatment on certain measures over time, and importantly, to monitor the duration of any effect that we see. And what you see in this chart is the difference in letters read on an ETDRS chart, which is what you see in the bottom right-hand panel there. It's essentially I suppose, a Snellen chart that you see in opticians in reverse. And we're measuring the difference between the number of letters read in both the untreated eye and the treated eye. So what you're looking for here is a delta between those 2 measurements that's significant enough to warrant further investigation. And the numbers you see in light blue here in the panel certainly meet that criteria. And you're also looking for a strong enough absolute signal in the treated eye. And again, you can see that we're definitely seeing that. Importantly, what we're also seeing here is a very stable, measurable duration of effect. We do have 1 patient now at 18 months follow-up. It is only an N of 1, so one has to be rather circumspect regarding what we're seeing in the graph on the right-hand side there. But nonetheless, very encouraging data, and certainly data sufficient enough to warrant progressing further with this study. And to that extent, as I mentioned, we now have regulatory approval to open out this study under a slightly amended protocol, both in the US and also into Europe, including the UK. We're going to be treating an extra 9 patients. We're going to escalate the dose from 1 million to 2 million cells to see if we can amplify further the efficacy signal we're already seeing. And we know the cells are safe at that dose level, so we may as well go for it to see if we can get an emphasized efficacy signal. And we're also enhancing some of the ways, not only that we get the cells in terms of the surgical technique to get the cells to the back of the eye, and also enhancing some of the efficacy readouts and measures that we're using in this study, including microperimetry. So in terms of the next steps of this program, it's all about generating further phase 2A data in those extra 9 patients, and of course following up the patients we've already treated to see if we can maintain that duration of effect in the 10 patients we've treated in phase 2 so far. And certainly if we can replicate the data we're seeing so far in a pivotal study, then we really have something of interest here. And to that extent, if the data does hold in phase 2A, we expect to be able to file for a single pivotal study before market application. And we're hoping to do that at some point in the second half of next year. So again, some really important data coming through on this study, and something that we can continue to update the market on as we treat those extra 9 patients over the next 12 months or so. So now I'm going to move on to the exosome and iPSC programs very briefly. Just a quick sort of science lesson on exosomes, and I'm a non-scientist, so I'm not going to spend too much time on this, obviously. But exosomes are essentially vesicles, lipid-based vesicles that are secreted by most cell types. It was thought historically that they were secreting cellular junk, essentially, but now it's known that they play a key role in intercellular communication. And we've been developing our own exosomes from our neural CTX cell line for a number of years now. And our aim, as I mentioned earlier, is to use our exosomes as a potential drug delivery vehicle to deliver complex novel drugs on a collaborative basis. And it's certainly true that there's increasing industry interest in the commercial value of exosomes for that purpose. Importantly, we believe that our own exosomes derived from our own proprietary cell line have a number of advantages, distinct advantages. As I mentioned earlier, we know that they can cross the blood-brain barrier. Perhaps that's intuitively understandable given that they're originally derived from a neural or a brain stem cell line. We can load small RNAs, proteins, and other agents onto these exosomes. Importantly, we can harvest the exosomes, purify them, and characterize them under GMP conditions. Bear in mind that they're derived from a cell therapy candidate that itself is manufactured under those same conditions. We have established analytics for all of that. And importantly, we can also modify our exosomes to engineer them to target particular tissues. And we have presented data with our collaborators, our academic collaborators, over the last couple of years that build on some of the notions that you see on this slide. So what that means is it opens up a broad sweep of opportunities, if you will, for the use of our exosomes to target a range of different disease conditions, essentially by enabling the exosomes to express certain factors that we can attach to them. So we know that they can express BDNF, for instance, a neuroprotective growth factor, if we're targeting exosomes against neurodegenerative diseases in the brain. We also know that we can deliver small RNAs against the KRAS G12D mutation, which is prevalent in glioblastoma and pancreatic cancers and so on. We also have a candidate that expresses the SARS-CoV-2 spike protein for potential delivery of antigens against COVID-19, most especially to stimulate an enhanced T-cell response in that condition. And this chart also shows you a couple of potential uses for our iPSC platform as well. So this is slightly different. This is using CTX cells essentially to derive induced pluripotent stem cells, essentially by reprogramming CTX into an embryonic stem cell state and then differentiating those embryonic cells or those induced pluripotent cells down into new cell lines of interest. It might be a hematopoietic cell line for allogeneic T-cell production, or a pancreatic progenitor targeting diabetes. So again, we have a couple of nascent programs to explore our iPSCs in those indications. And I spoke about the potential commercial interest in exosomes. And this slide just gives you an indication of some recent deals that have been done by other players that are focused more, almost exclusively actually, on exosomes in their businesses. And Kodiak and Evox in the US and UK, respectively, are the two players that we watch. They're the ones to watch in this field as exclusively exosome-focused companies. And you can see that even at a relatively early stage in this field, there is real deal value potential here. And what we're focusing on is generating early proof of concept in collaboration with third parties to see if our exosomes can add value to those third-party drugs, and subsequently, if that's right, if that's positive, to generate license deals with those third parties. So at the moment, as I mentioned earlier, we've signed 2 or 3 collaborations with others in the pipeline, and ultimately, if we can get some hits, that will lead to higher value licensing deals in due course. Finally, our induced pluripotent stem cells, again, I mentioned that this is essentially reprogramming CTX cells into a pluripotent state, which we can then derive new cell lines for. And importantly, what we've learned and presented actually in conference is that when we actually produce those new cell lines by differentiating our iPSCs, those new cell lines maintain the immortalization characteristics of the CTX cells, immortalization being the ability to expand these cells indefinitely. So essentially, it's an engineered cell line without any need to rederive from fresh tissue to generate more cells. So really, we think our iPSCs have great potential in generating new therapeutic candidates, new cell lines that can be used to target particular disease conditions using certain new cell lines for that purpose, such as T cells for CAR T therapy and so on. So again, it's early stage, but something that we see as a second wave of potential value generating programs for this business. Finally, just a quick word on our CTX cell therapy platform. Again, CTX is a neural cell line. It's the one that we've been working on the longest. It's in the clinic against stroke disability. It has an excellent safety profile. We've published phase 1 and phase 2 clinical results with this candidate. Again, it's commercially attractive in that we can scale it very, very effectively. It's an engineered cell line. Again, it's formulated in a cryopreserved fashion for easy shipping and storage. And currently it's licensed for development in China. And we decided and announced a couple of weeks or so ago that we would now pursue a licensing strategy for this particular program. So going forward, our in-house efforts will be spent exclusively on the retinal program for retinitis pigmentosa in terms of our cell therapy programs. The stroke program will continue, but under license. And as I mentioned, we also intend to license CTX for use in other neurodegenerative conditions for parties that are interested in doing that. And Huntington's disease is where we have some recent interesting data in that regard. So just to summarize then, we're a global leader in cell-based therapeutics. We have presence in the UK and in the US. We have allogeneic or non-patient specific cell platforms. These are scalable, they're patented, and give us the opportunity to generate cell-based therapeutic candidates in a cost-effective way. We have exciting early stage phase 2A data in RP, and we're looking to generate and build on that data over the next 12 months. If it continues to be as positive as it has been, we see a real value inflection potential in this program as we garner those data. And as I mentioned, there's an increasing level of industry interest in exosomes, so we're exploiting the potential of our own platforms in that regard as well, in a collaborative way that, as I mentioned, will lead hopefully to higher value licensing deals as we go forward with that program. And with that, I'd be happy to take some questions. Thank you.
So I'm going to only have a chance for a couple of questions, I'm afraid, and so I'll try and sum up some of the key points we've had questions about. And cash position, where are you at the moment? Are there any milestone payments due from Fosun in the next, I guess, 12 months or so? If you could talk around that, please.
Yeah, sure. Actually, we announced our prelim results at the end of March yesterday, and we went into some of those details yesterday. But our cash position at the end of March was 12.6 million. That gives us about a year's worth of runway from where we are now. There will be further milestones from Fosun. It is, of course, dependent on Fosun making progress over the next 12 to 18 months with both of those programs, the retinal and the stroke program in China. But there are milestones due if they do make that progress in terms of garnering regulatory approvals, for instance, in China, to pursue those programs at a clinical level in that territory. So yeah, there will be some more milestones over the next year or two. We have cash on hand, obviously, at the end of March that gives us a runway, notwithstanding that.
Thank you. The HRPC platform, where you've been working on RP, are there other conditions that could be benefited by this?
Yes, as I mentioned, we do see the HRPC as a platform, so we believe it will have merit beyond RP. One example might be cone rod dystrophy, which is sort of the reverse of RP. It's a loss of central vision first, but very closely related to RP. And macular degeneration could be a candidate as well. We don't have clinical data to support HRPC in those indications, but certainly there would be merit in trying to pursue those. Again, if we can show good proof of concept clinical data, continue to do so in the first target we've chosen, which of course is retinitis pigmentosa.
Okay, and then finally, a name from the past a little bit. Neil Woodford showed great faith in the company and was a large shareholder. Has that now gone to Schroders? Is that still in the trust? What's the current state of play with that, please? Yes,
actually, so there were two aspects to that. So we had a holding, or Woodford had a holding in us in both the old income fund and the trust. As you rightly say, the trust part of the holding has now gone to Schroders. We know Schroders quite well. We've met them. We believe they are perfectly comfortable with the story and holding the stock, so that's great. The other half, the income fund stake, went to a German family office called Obertrischer Capital, who have probably done more due diligence on the company than virtually anyone else that we know, certainly in recent times. And so they took out Woodford's entire stake in the income fund, so about 16% on the register. And again, we're delighted to have Obo as a lead shareholder for us. We know them well, and we very much hope they will continue to support the company, ultimately to get to what we truly believe is a very exciting end game with these programs.
Lovely, Michael. Thank you very much indeed. Thank you for joining us. Thank you for presenting.